Welcome to another edition of CCSD Living Environment. Today we're going to look at structure and types of RNA, and we're also going to talk some about transcription. First of all, if we're going to talk about RNA, we need to have this understanding that RNA is very similar to DNA, obviously by the name. So RNA is going to be ribonucleic acid. So that's RNA. RNA is a polymer made of a smaller repeating subunit, or monomer, known as a nucleotide. Now, hopefully right away you're saying, okay, there is a similarity to DNA because we said the same thing about DNA. DNA is a polymer made of nucleotides. So you have DNA nucleotides and you have RNA nucleotides. So let's look at the difference between the nucleotides. In an RNA nucleotide, they have a 5-carbon sugar called ribose instead of deoxyribose. Again, they have a phosphate group, and they have a nitrogenous base. So in RNA, you have adenine, cytosine, guanine, and this time we're going to have uracil instead of thymine. So there's still 4 nitrogenous bases. The difference comes in that thymine is replaced with uracil. RNA is a single-stranded molecule. So instead of that ladder that we had for DNA, imagine cutting the ladder in half and only being left with half of that ladder. That half of ladder that you're left with is a molecule of RNA. It is single-stranded. There are 3 different types of RNA. The first one is called rRNA, or ribosomal RNA. It is formed in the nucleolus of the cell, and it binds with ribosomal protein in the formation of ribosomes. Remember, ribosomes are the part of the cell where proteins are made. And then I have mRNA, or messenger RNA. Messenger RNA carries the instructions, or the blueprint, from the DNA molecule to the ribosome. So the messenger RNA is exactly that. It is the messenger. It's carrying that message, those instructions, from DNA out to the ribosome in the cytoplasm. And then I have transfer RNA, or tRNA, that's going to bring the amino acid or transfer the amino acid from the cytoplasm to the ribosome. So you can see here, hopefully, that we're trying to go from DNA to a protein that's being made in the ribosome. And the transfer RNA is transferring those amino acids in for us. So how is the RNA actually made? The RNA is actually made in a process called transcription. Transcription is a process where we make RNA. During transcription, the enzyme RNA polymerase, so RNA polymerase, again I know it's an enzyme, it ends in ase, it's the enzyme that makes the RNA polymer. So the enzyme RNA polymerase uses one strand of the DNA as a template to assemble RNA nucleotides into a strand of RNA. So again, we're using the DNA as a template, and we're going to be complementary. But remember, instead of a T in RNA, we have a U. So let's take a look. Here's my strand of DNA. Here's my strand of mRNA. Notice, A this time is going to go with U. T goes with A, G goes with C, C goes with G, G goes with C, T goes with A goes with U, C with G, G with C, T with A with U, C with G, A with U, G with C. So the process is exactly the same as it was in making DNA from DNA in replication, but in transcription, when we're making RNA, instead of putting in a T, we put in a U. In the nucleus, the enzyme RNA polymerase separates or unzips a section of DNA, exposing the base pairs. mRNA is then synthesized with RNA nucleotides are matched to the complementary base on the DNA template. The instructions or blueprint has been copied or transcribed from DNA to mRNA. Many of you at this point along the way have read Shakespeare in English class, and you're probably familiar with this idea of Old English versus English that we speak today, or maybe the English that they speak in England versus what we speak in America. So the idea then of having DNA and RNA, they are very similar. They're both made of nucleotides. I'm going from the language of nucleotides to the language of nucleotides. That process is transcription. If you go from Old English, what Shakespeare wrote in, to the English we speak today, we would transcribe his literature. Because it's the same language, it's just a little bit different. It's still the language of nucleotides, it's just a little bit different. The RNA polymerase will only bind to a specific part of the DNA, and that specific part of the DNA that it will bind to is called a promoter. So there's a region on the DNA that will actually allow RNA polymerase to bind to it so it can be turned into mRNA. That signal will show the RNA exactly where it's going to start being made. So RNA polymerase can come all the way down the DNA molecule, and what will happen is it'll get to a point where it says, hey, this is where you're going to start making the RNA. So the RNA polymerase will bind on, and then it will actually start making the RNA. There are similar signals in the DNA that cause the process of transcription to stop. So the RNA polymerase will be going along reading the DNA, and then there's a signal there that says, okay, you're done. This is all you need to copy. This is the only part that we actually need to turn into a piece of RNA. So the whole process will stop. If you've ever been to a movie, which I'm sure you have, or you've watched a movie, or you've watched a TV show, you know that editing needs to take place from what they originally filmed to what finally gets released for us to watch. Well, some editing needs to take place with the RNA also. The editing that needs to be done is so that the RNA molecule that's leaving the nucleus is actually the RNA molecule that we want to turn into the protein. That's the molecule that we actually want to go out to the ribosome. Because the DNA may have some other stuff in it, maybe it's a protection thing, maybe it's something else, but there's some stuff in the DNA that we don't necessarily want to have in the RNA. So the parts that are cut out are called introns. Those parts are going to stay inside the nucleus. They're not going to leave. They're taken out while they're still inside the nucleus because remember, that's where the DNA is. So this process of DNA going to RNA takes place in the nucleus of the cell. If it's a eukaryotic cell, like we are, that has a nucleus or nuclear membrane. The pieces that are left are called exons. They are spliced together to form the final mRNA that is going to exit the nucleus. So the exons are what exit. And that gets a little bit confusing because you would think exon would be what you're getting rid of. But the way to think about it is, what is the part that's actually going to leave the nucleus to go to the ribosome? The exons are going to exit. The introns stay in the nucleus. So what does this whole thing look like? What is this whole idea about editing this RNA? What I've tried to do is give you a picture similar to what's in your notes. So what's in blue are my introns. Those are the pieces that we're going to cut out. So if we take all of the DNA that you have, think about that for a minute. Do you think every single piece of your DNA actually codes for protein that makes us who we are? Well, no, of course it doesn't. There's a whole bunch of DNA in there. Maybe it's there to protect the good parts of the DNA. Maybe it's just stuff that's left over from evolution of humans. But there's some stuff in there that we don't necessarily need. So we're going to cut those introns out, and that's going to leave us just with those exons. So you can see here my final piece of mRNA that's going to leave the cell is smaller, and it only has the exons in it. So on the slide before, I mentioned something about splicing. If you've ever taken two pieces of wire and connected them together, that's splicing. Way back when we were kids, we had things called cassette tapes, which you may or may not be familiar with. But with a cassette tape, if you took the tape and you cut it and you taped it back together, the tape would still play in your tape player. When you go to watch a movie before the digital age, it used to still be on a film strip. And what would happen with a film strip is they would take one part of it and they would connect it to the other part. And whenever you're making a connection like that, it's called splicing. So with gene splicing, or with the splicing of RNA, we're taking this exon and we're going to connect it to this exon, to this exon, to this exon. So splicing is cutting out the introns, which is the part of the DNA that's not going to code for a trait, and we're going to leave the exons in the final piece of mRNA. So the idea of splicing is to basically take a couple of things and hook them together. And that's the idea behind gene splicing. So this has given you a little bit of an idea how we start to go from DNA, the instructions in the cell, to actually flesh and blood, to what we are, which basically is all protein. We're a big walking bag of protein, as we've talked about before. Whether it's hormones or antibodies or pigments or enzymes, whatever those proteins are. So in the next one, what we're going to look at is how do we actually go from taking this piece of mRNA, how do we actually take those instructions from the DNA out to the ribosome and make a human being, make a dog, make a cat, make an elephant, make a maple tree. That's the next step in our process. Thanks for watching, and I'll see you next time.